Hello, everyone, and welcome to the Kids First webinar for the X01 pre-application discussion. My name is Laurette Javoy, and I'll be doing the introductory part of the webinar. I'd like to just go over a few of the instructions so that you understand how we're going to operate the webinar. This program is being hosted by the Gabriella Miller Kids First Pediatric Research Program, and several of the people involved in the program here at NIH will be talking with you today. Right now, all the participants are muted upon entry to the webinar, and we ask that you please remain muted until the question or discussion period at the end of the presentation. At that time, you can unmute yourself by selecting *6 on your telephone or clicking on the mic symbol under the audio connection on the webinar. You can ask technical questions at any time using the chat service through the webinar to the host. If you are having technical difficulties, please feel free to use this means of communication. Please save any questions related to the program or the funding opportunity announcement for the question period at the end. And if by chance you have additional questions after the webinar is over, please feel free to email them to kidsfirst@od.nih.gov. And just a reminder, we are recording this webinar, and the recording will be posted to the Kids First website in case you want to review any aspects of it or you have colleagues that were unavailable at this time to listen to it live. So this is the pre-application webinar for the discovery of the genetic basis of childhood cancers and structural birth defects, the Gabriella Miller Kids First Pediatric Research Program X01 initiative. And before we get into the details of this funding opportunity announcement, I want to provide a little bit of background information and introduce to you the people that comprise the Kids First leadership group here at NIH. We have a number of program staff involved in this Common Fund project. As you know, it was initiated in 2015 with the overall goal of establishing a Kids First data resource for the pediatric research community. We're very excited by this opportunity, both for NIH and for the pediatric research community. And the program is focusing on structural birth defects and pediatric cancers. Since this is a Common Fund program, leadership responsibilities are being shared across 4 NIH institutes as well as the Common Fund. And the people listed on this slide are the folks that are involved in coordinating the program. As I mentioned, I'm Laurette Javoy. I'm a program officer at NICHD. Jonathan Kaltman is from NHLBI, and he and I are the points of contact for structural birth defects questions. Adam Felsenfeld is at NHGRI, and along with James Coulomb from NICHD, they're the point of contact for sequencing questions. James is the project officer for the Kids First Sequencing Centers. Malcolm Smith and Jamie Goudry-Alville are program officers at the National Cancer Institute, and they're the points of contact for pediatric cancer questions. And Martin Lurkes is involved with our data resource project, which is soon to be funded this year, and he'll be the project scientist coordinating that effort. From the Common Fund, we have Marie Nereus, who is our program officer, and Mike Steenstra, who is the operations and budget person. We also have a person who handles policy planning, evaluation, and communications for us. That was Joe Tiano, who's recently left and moved on to another position here at NIH, and we're in the process of getting someone new. And you all are probably familiar with Valerie Cotton, who is our administrative point of contact. She's the one that coordinated your participation in this webinar today. I just want to take a moment to point out that there's more than the usual interest in this program from both Congress and the advocacy communities, partly because of the way the program came to be. And today we have Stephanie Couchet from the Common Fund on the line with us, and she's the team leader for the Common Fund's Policy Planning, Evaluation, and Communications Office, and she's filling in the role of the communications person today during the webinar. So if you have any questions from this aspect of the program, she'll be able to answer them during our discussion period. So the people highlighted in bold on this slide, myself and John Kaltman and Valerie Cotton, will be the people talking with you today during the course of the webinar. Okay, on the next slide, I just want to go through a bit of background as to how we got to where we are today. So the program was initiated in response to the 2014 Gabriella Miller Kids First Research Act, which was signed into law on April 3rd, 2014. This law actually did three things. It ended the taxpayer contribution to the presidential nominating convention. And if you look at the small graphic at the bottom of the slide, this is a snap of the upper portion of your IRS 1040. And the area highlighted in red was actually a little checkbox that existed on the 1040 back in years prior to 2014 and 2014, which allowed taxpayers to contribute $3 toward this presidential nominating convention's fund. It turned out a lot of money was sitting in this fund and wasn't being spent. So at the time this law was passed, what it did was it authorized the transfer of $126 million, which were sitting in this fund, into a new Pediatric Research Initiative Fund. And it also authorized Congress to appropriate $12.6 million a year for each of 10 years directly to the NIH Common Fund for a new pediatric research program. And the first appropriation was accomplished in fiscal year 2015. I think from our perspective, in running this program, the most important word on this slide is it authorized the appropriation on an annual basis. So it's very important to remember that this program, there isn't a mandate for 10 years of funding. We're dependent upon congressional appropriation on an annual basis to get the funds for this program, which harkens back to my earlier statement that there's a lot of interest on the part of advocacy groups to see that this happens on an annual basis. Okay, so if we look more closely at the Kids First Working Group, as I mentioned, it's a trans-NIH effort supported by the NIH Common Fund. The institutes that chair the Kids First Working Group are the Child Health Institute, Heart, Lung, and Blood, the Cancer Institute, and the Genome Institute. But it's very important that you understand there are a number of other NIH institutes working on this program, and they're listed here on the slide. So just to briefly reiterate, it's the Dental Institute, the Drug Institute, Alcohol, Diabetes and Kidney, Environmental Health Sciences, the Eye Institute, Arthritis and Musculoskeletal Diseases, Infectious Disease, and the Office for Research Infrastructure Programs. And we also have an ex officio member from the CDC's Birth Defects Division sitting on the working group. So we've got a lot of people involved in this program, and we want you to be aware of this because if you're interested in applying to the X01 program, it may be that your mission aligns more with one of these other institutes, and you should feel free to contact program officers you know at these institutes if you have questions about how relevant what you want to do is to their mission. For a complete list of our working group members, you can go to the Kids First Common Fund website and look at working group members at the link there. And now I'm going to turn the webinar over to John Kaltman, who's going to review the program goals.
All right, thank you. So the overall goal of the Kids First program is to develop a data resource, which we're calling the Gabriella Miller Kids First Pediatric Data Resource, where the pediatric research community can access well-curated clinical and genetic sequence data that will allow them to identify genetic pathways that underlie childhood cancer and structural birth defects. A greater understanding of the genetic underpinnings of these pediatric conditions is critical for developing and improving preventative measures for diagnostics and for therapeutic intervention. There are three main initiatives that support the Kids First program. They include cohort identification and DNA sequencing, the Kids First data resource, and data analyses. we'll talk about the first one mostly today, although I'll briefly mention what the other two will be doing as well. Cohort identification and DNA sequencing is accomplished through the X01 program, which will be the topic of discussion today. The goal is to identify cohorts of children with either childhood cancer or structural birth defects, or both, and their families for whole genome sequencing. Kids First will support the whole genome sequencing of these cohorts to elucidate the genetic contribution to childhood cancers and the genetic etiology of structural birth defects. The Kids First Pediatric Data Resource will be awarded and established later this year. The goal of this resource will to develop a web-based portal that will aid researchers in identifying cohorts that are available and that have sequence data, and that will also facilitate cross-cohort analyses. And then finally, data analyses initiatives, which are further down the road, will be set up after the data resource is up and running, and will further support analyses of Kids First generated as well as non-Kids First generated data to uncover new insights into the biology of childhood cancer and structural birth defects. So now to talk about the X01. This is the third cycle of our X01. It's currently called Discovery of the Genetic Basis of Childhood Cancers and of Structural Birth Defects. The first one was released in 2015, and through the 2015 X01, 2 childhood cancer cohorts and 5 structural birth defects cohorts were sequenced. In 2016, 3 childhood cancer cohorts and 5 structural birth defect cohorts were sequenced. And then the current X01 is FY17-063. We hope and plan to continue sequencing for several more years, provided that funds are appropriated, as Lorette discussed, and that cohorts are available. The funding announcement has slightly evolved over the 3 years of the program. In particular, less of an emphasis is being placed on the minimum number of samples. Also, less of an emphasis is being placed on trio design, as long as there is a valid analysis plan that accompanies the cohorts being proposed. So just a word about the X01 mechanism. The X01 is not a typical award mechanism or a typical grant mechanism. There is no award that's given to the recipients, and in fact there is no notice of award. Recipients are selected for the opportunity to have their cohort sequenced. Again, no funds are provided for analysis, but there is an accompanying R03, which is PAR16-348, which originates from NICHD but has a number of partners that supports analysis of data that is generated by the X01 project. Also, these awards are not listed in the NIH reporter, but abstracts from our prior awardees are listed on the Kids First website, and the web address is on your screen. So now I'll turn it back to Valerie.
So I'm Valerie Cotton, and as Lorette mentioned, I am the program analyst for the Kids First program, and a large part of my job is to liaise with the sequencing center staff, as well as the X01 investigators who will be selected through PAR17-063. I'm going to go over some details that will hopefully help you with your application to this announcement. And some of the information I'm about to cover is already addressed in our FAQs. If you are considering applying for this at all, I highly encourage you to look at our FAQ page, which you can find on our Kids First Common Fund website at this link here. We are constantly updating the FAQs. We'll update them after today based on the questions that we get and the discussion that we have. And we update them on an ongoing basis as we get specific inquiries. So these are the sequencing centers that are providing the sequencing services described in the X01 FOA. They were selected through a cooperative agreement, and we will be working with these centers for 3 years. Starting in fiscal year 16, those cohorts have already been selected, and the samples are being shipped, and they're entering the sequencing pipeline around now. And we'll be working with them through fiscal year 18. The first center is the Hudson Alpha Institute for Biotechnology, led by Sean Levy, in partnership with St. Jude Children's Research Hospital, led by Jingwei Zhang. The other center is the Broad Institute of Harvard and MIT, led by Stacey Gabriel. Both of these centers have the capacity to sequence both cancer projects and birth defect projects. However, our plan is to assign all cancer projects to the sequencing center at Hudson Alpha St. Jude to take advantage of the analytical expertise for identifying somatic cancer variants that was offered by the center. But depending on the applications we get, the approaches that are proposed, the size of the cohorts we select, the assignment of projects may not work out exactly this way, but this is our general framework. The sequencing centers are tasked with performing the sequencing as well as the variant calling of already existing DNA and RNA. So that means that they'll generate the variant calling files as well as BAMs and in some cases FASTQ files. Birth defect cohorts will receive whole genome sequencing at an average of 30x coverage. Cancer cohorts will also receive 30x coverage for whole genome sequencing for germline samples. But one change in this FOA is that we are really now encouraging the submission of nucleic acids from tumor samples to go to the Hudson Alpha St. Jude Center, where they would generate whole genome sequencing as well as whole exome and RNA sequencing, because this is the information they need for their analytical pipeline. You may propose other methods or technological approaches for sequencing in your applications. For example, this past cycle, someone proposed higher coverage than 30x. In the end, after working with us and their sequencing center, they determined that 30x was the most reasonable. They wouldn't have to decrease their sample size if they wanted higher coverage, and they decided that the higher sample size was more valuable. So this is just an example of how you can propose other things, but the final project design will be decided among discussions with X01 investigators, sequencing center staff, and NIH program staff. Another possibility that you might be interested in proposing is linked long-read sequencing. This is actually a technological approach that was proposed by both of these sequencing centers. But since this is a new technology, we've actually asked them to work together to run a small pilot study. And this is so that we can evaluate and better understand the added value, the utility of this approach for these types of cohorts for birth defects and pediatric cancers. They're doing this using the 10X platform. So based on the results of the pilot study, this is something that may become more available for future opportunities, including this one. So if this interests you at all, I encourage you to describe this in your application and to provide a justification for why you think it might be useful to your particular cohort. I just want to take a moment to talk about where data lives and how it's accessible. So all Kids First studies will have to be registered in dbGaP. If you're not familiar with dbGaP, it is NIH system for facilitating controlled access of genetic data to other researchers. So you would be the primary researcher generating the data, but a goal is to make this data accessible to other researchers as well. And dbGaP is sort of the gatekeeper of that process. So birth defect projects will all be registered in dbGaP, and phenotype data will also be submitted to dbGaP. Sequence data will be submitted to NCBI Sequence Read Archive. Actually, the Sequencing Center will be responsible for this, but I just wanted to make sure you guys knew where data would be living. And for childhood cancer projects, they also have to be registered in dbGaP, but all data, biospecimen, phenotype data, and sequence data will be submitted to NCI's Genomic Data Commons. This is a database that was launched within the last year, It's still a little bit new, so NCI staff is working with us and our investigators and sequencing centers to make sure that we get the necessary information from each project properly submitted. So now I'm going to address a few of our expectations or requirements in terms of what Kids First program goals are. So from the beginning, we've been very focused on whole genome sequencing to discover genetic variants contributing to these pediatric conditions. But we've added the opportunity for whole exome and transcriptome sequencing for tumors as well, based on the proposal and the value of this argued by Hudson Alpha St. Jude. So given that these are the sequencing technologies that would be performed, our expectation is that the DNA, and in the case of tumors, RNA as well, extractions that you plan to submit are of sufficient quality and concentration for these technologies. We'll talk a little bit later about how we ask you for that information in the application. Another goal of our program is to facilitate cross-cohort analyses and this needs to be done based on common data elements. One very large task of our upcoming Kids First data resource will be to harmonize phenotypes across all of our projects. So to this end, we really need at least some basic data elements, but the more detailed phenotype data you have, the better. For birth defect projects, we don't have an established set of standards for phenotype data, but since the cancer data will be going into NCI's Genomic Data Commons, they have already clearly established the data elements that they expect for biospecimen and clinical and phenotype data. So you can find this link here and also in our FAQs to look at the GDC's data dictionary. Another goal of ours is program balance. So we seek to broaden the diversity of both pediatric cancer and structural birth defect data sets that will eventually be part of our Kids First data resource. This means that if we get a lot of applications, and we hope that we do, and we are faced with this choice of two very similar applications, but one proposes a sequencing of a condition that we haven't covered before, we are more likely to select that cohort. This doesn't mean we discourage you from applying if you have a cohort that's similar to what we've done before, but we did want to be transparent about this. And we, in previous slides, gave you a link to where you can find the list of cohorts we've sequenced before, and we encourage you to look at that. A huge goal of ours is data sharing, so to make data generated by our program as accessible to the research community as possible. So in line with NIH's genomic data sharing policy, cohort participants must have given consent to allow the sharing of individual level sequence and the associated phenotype data through an NIH-approved repository, the classic example being dbGaP. Another priority of ours is to make sure this data can go to as many different projects and researchers as possible. So we are most interested in data that is consented to be shared as broadly as possible, so something that is consented for general research use, something that is consented for health, medical, or biological research is preferred over disease-specific data use restrictions. And it's just because we want to promote collaboration among investigators as much as possible, and this just becomes a little bit more complicated when data sets are restricted to certain researchers. Again, it doesn't mean we discourage you from applying if you have a disease-specific restriction, but we do ask that you communicate that in your application, and some of you might be in a position to start recruiting new cohorts, or you might be reconsenting for another reason anyway, and NIH has guidelines to guide you to have language that allows for broad sharing. Sample size and family structures. As John mentioned, in previous years, we've really focused on a trio-based design, but we realize that there are other family structures and designs out there that can lead to genetic discovery, but this needs to be very clearly communicated in your application. Obviously, the larger the sample size, the better. In some cases, in the birth defects, in the FOA, we talk about the birth defects minimum being 100 trios, but if you have less than that, we would just like to see a clear scientific justification for how you will be able to identify variants. If your sample size is not that large, we strongly encourage you to consider collaborating with other investigators to pool samples together for a larger sample size. And of course, in the case that you don't have trios, you just need to show us an argument for how that you have enough probands, that you will sequence enough affected and or unaffected family members to lead to genetic discovery. And this ties in very closely with analysis plans. So the goal of asking for analysis plans in these applications is to have investigators demonstrate that the proposed project has an adequate research design for genetic discovery, and that you as the X01 investigators are in a position to perform these analyses. So one modification of this iteration of the FOA is that we do communicate that we recognize the analytical power of each data set will increase once it's incorporated with all the other data sets that are part of Kids First. Nevertheless, it is important that you demonstrate the data will be usable on its own. If you feel that your team does not have the analytical or statistical expertise to perform these analyses, we highly encourage you to partner with another group who may be able to work with you on an analysis plan. So now I'm going to move on to address a part of the funding announcement called Other Attachments. In prior years, we've always discussed the institutional certification in this section, but for this year, we've greatly expanded this part of the application to ask for sample information, a description of the phenotype data, and a description of the family structures. And the reason we ask for this here is that we want to enable you to submit supplemental additional material to fully explain and respond to the information we ask for here without really cutting into your limited research strategy. So we have also created an optional downloadable form that is available on our FAQs that is a template where you can give us the information that we're asking for. This is optional. You may choose to respond to this information that we need in another format, in whatever way is best for you. But the form just serves as a place where both applicants and reviewers can look at and work with a uniform document where the information can be organized. And I'm going, in the next slide, I'm going to go over each of these one by one. So the first attachment would be the institutional certification or a provisional certification or a sample consent form. Institutional certifications are documents that assure that the genomic data meets the expectations of NIH's genomic data sharing policy. Many teams cannot usually get a full official institutional certification from their IRB until after an award, or in this case, after you've been selected for the X01 program. So you may provide the provisional certification or the sample consent form instead. Please, for the certifications, please use the current NIH templates, which could be found at this link. That's where you can find both the institutional certification and the provisional certification template. No matter what you submit, it just needs to be clear. You need to cover all the sites that contribute samples to your project. It needs to specify whether data is consented for deposition in dbGaP or another NIH-approved repository, and the data use limitations need to be very clearly explained. This snapshot below is an image from the institutional certification, so you can see how ultimately you will have to organize and lay this out for us. But if you're not submitting a full institutional certification with your application, you need to describe them somewhere else in the application. And I just want to mention that no matter what you submit with your application, if your project is selected, you will have to provide the full institutional certification using this template shortly after we notify you that your project has been selected. And this is the first step of that dbGaP registration that I talked about. We're not going to get into those details here. Just know that NIH staff is here to help you with this, but this would be that first step. The other attachments we ask for are sample information. So we really need to understand the total number of samples that you are proposing for sequencing very clearly. Again, you can modify this table. You can submit the information we ask for whatever way works for you, but this is one way you can organize these details. So this table, the rows are organized by sample type or DNA or RNA source. The top part of this table would be for both birth defects cohorts and cancer cohorts, and the bottom part is for tumor samples for cancer projects. We just need you to describe the number of samples from each, the extraction method that was used, the concentration, some metric of quality, and then tell us which metric was used, method of quantification, and then we need to know how many samples will be ready to ship by August 2017. In a few slides, I'll talk more about the timeline of this program, but we anticipate that we will be notifying who we have selected under this X01 fiscal year 17 iteration in July. So we are hoping that we will be getting data, samples in the pipeline by August. But we realize that sometimes there are some lingering extractions to still finish up, and sometimes there are some shipping logistics to coordinate. So we ask for how many samples are ready now and how many samples are ready 6 months from now, just to get a sense of what is ready to go. Again, you can modify this slightly. If you know that you'll have everything ready by November of 2017, then you can change the January 2018 to November 2017 and tell us when all of your samples will be submitted by. We also ask you to describe what phenotype information and demographic information you've collected from these participants. At minimum, we expect age at diagnosis, maybe you have some other age information, sex, race, ethnicity, and then just tell us what else you've collected. Describe the primary clinical description, the type of birth defect, the primary tumor site, and tell us what other phenotype information, whether it's clinical or otherwise, that you've collected. We're also interested in knowing what family medical history you have. This is a very general table. Again, we just need you to give us a sense of what phenotype data you have available. Again, for cancer cohorts, this is a little bit more clearly laid out in the standards provided by the GDC, so we highly encourage you to look at those. And again, for those cohorts that are non-trio designs, we ask that you describe what the family structures are. You might have a proban parent-sibling setup. You might have multiplex families. You might have consanguineous families. We just ask that you submit enough information to make it clear how many samples per family you're planning on sequence and whether they're affected or unaffected. In many cases, it might just be simplest to submit pedigree information, but again, tell us who you're proposing for sequencing. If you don't have pedigree information, we ask that you just find another way to really make it clear what you're proposing. This is an example of a table that made sense to work from with one group. They had some trios, but they also have situations where a proban and another first-degree family member was affected with some unaffected family members. So we just need it to be clear how many of each family type, if you have many different family types, and how many samples from unaffected and affected family members you're proposing to sequence. A couple other things that didn't quite fit into other slides. We just want to emphasize that another change to this iteration of this program is that we are encouraging the submission of applicants who have already sequenced a case or a proban through other funding, but have unsequenced nucleic acids from a parent, from siblings, from a tumor. If you do this, we do ask that you pretty clearly describe the sequencing technology that was used on the case of the proban, but we are certainly open to these kinds of proposals. We also want to emphasize that this opportunity is open to the NIH Intramural Research Program, and that includes collaborators of the NIH Intramural Research Program. And that's because this doesn't come with formal funds. It is that opportunity for sequencing. So for this cycle, the 2017 cycle, you may have heard me talk about a lot of things that we prefer and expect, but really, no matter what, we encourage any investigators that have a cohort that meets the minimum requirements to consider applying. As you saw, we're working with our current sequencing centers until 2018, but we also have plans to continue sequencing in later years. So we encourage you to start collecting samples that meet our requirements now, but stay in touch with us about this. Again, we'll continue the sequencing as long as funds are appropriated and cohorts are available. So we need to stay aware of what cohorts are out there. This is the timeline for this cycle. It is on an earlier timeline than our previous 2 years, so we wanted to make sure everybody was clear on this. This funding announcement was published in December. Today is January 23rd, and this is the webinar. The letters of intent are due February 7th. As you know, these are not required, but they do help us prepare for the review, so they're helpful. March 7th is the application deadline. We anticipate that the review will take place sometime in April, but we have not set a date. Between June and July, we will work with our working group to make a selection. Since there are no funds associated with this program, it doesn't go to an institute council. We work with our working group, and then we get a recommendation approved by the institute directors that co-chair our working group to make our decision. And then we plan to notify the recipients by July, and we hope to get these cohorts in the pipeline by August. So now I will open the floor up for questions. I just want to reiterate that you can continue to reach out to us. This is the email address for the Kids First mailbox, which we are monitoring. You also have the contact information for program officers who are leaders of this program in the funding announcement itself. My email address is on the slide deck, so we'll be publishing the PDF of this slide deck, as well as publishing the webinar, the recording of this webinar. And just a reminder, we'll take the questions now. We'd like you to ask them vocally. And so you'll have to unmute yourself. If you called in through a telephone, you can select *6 on your phone to unmute yourself. And if you called in through your computer, you'll have to go to the audio connection and select the mic symbol to unmute yourself. Sounds like somebody unmuted themselves as a test run. So we're ready to receive your questions.
Hey, thanks for the presentation. My name is Craig Wong, and I'm calling from the University of New Mexico. I had a question regarding existing cohorts that have been already uploaded to dbGaP, but with SNP level genotyping data. Does it make it simpler to have that already done ahead of time if we want to get whole genome sequencing for this data set? Go ahead.
I'm not sure I fully understand the question. Do you mean that you have a...
So I've already done kind of SNP genotyping and had already uploaded cohort data to dbGaP. However, we would like to get whole exome or whole genome sequencing for this existing cohort on that. And this is data, or this is DNA out of the DNA repository. So regarding the certificates and all the paperwork and registering for the, dbGaP and the kind of administrative requests that were presented earlier in the presentation, does that stuff get grandfathered in, or do we have to go ahead and, redo a lot of the paperwork?
You probably have to work with the folks who helped you register the original trial, but probably you can piggyback this data onto that trial. We've done that in other sort of scenarios that are somewhat similar, where a cohort has already undergone, for instance, exome sequencing, and then underwent whole genome as a part of this. So that data was sort of attached to the original cohort data, and obviously that will likely simplify the registration process if you've already gone through it and downloaded phenotype data as well.
Thank you very much.
Other questions?
Alex Rackett from Pittsburgh. I have a question regarding the phenotypes. Does it have to be one particular phenotype, or can actually the collection include several phenotypes?
Right. So, yeah, the short answer is yes, it can. Are you talking about birth defects or cancer?
Birth defects.
Or
I'm talking about birth defects.
Yeah. It certainly can. You know, part of the idea here was that from a genetic standpoint, we seem to find a fair bit of overlap between genetics and different phenotypes in birth defects. Obviously, I think an application would be stronger from an analysis standpoint if you can make an argument about how these different phenotypes might be analyzed together, but certainly that's possible.
And what about prenatal samples?
We're all looking at each other.
And that would be DNA obtained from what source?
It would be amniocentesis.
And would you have ultrasound data to support the fact that the fetus has a defect, a birth defect?
Yeah, that's the point, is that it would be, the phenotyping would be done by prenatal ultrasound, so it would be a very unique phenotyping data set.
It's not precluded from being submitted. We haven't had anything, and I guess we hadn't really considered the possibility of something prenatal. There would have to be some scientific justification to why we would devote resources to a prenatal cohort as opposed to a pediatric cohort.
Yeah, I can imagine reviewers might wondering about the sort of validity / accuracy of prenatal diagnosis, which obviously could potentially be bolstered by postnatal confirmation of that diagnosis. But, you know, I think you'd have to make a strong argument for why it's being done in this way as opposed to, as Lorette said, waiting.
Thank you.
Daniela, University of Washington. I was just wondering if there's a minimum.
Daniela, you're really quiet. I wonder if you're not close to your microphone or if you can find another way to make yourself louder.
Sure, I'll try.
That's better.
Okay. So I was wondering if there's a minimal concentration for DNA.
Minimal concentration.
Yes, if you go to our FAQs, we have some information about what our sequencing centers expect. If you want more details, feel free to contact me, Valerie, or the Kids First mailbox, and we can talk about that. But there is some information on our FAQs.
Thank you. And my other question was about the results. So when do you expect we would get the results back? So, and are they going to be VCF files, or are you going to send us the BAM files?
Yeah, so the sequencing centers will generate VCFs and BAMs, and those will be somehow transferred back to the investigator, and the investigator will have 6 months to work with their own data before it gets released to the public through dbGaP. So yes, we have ways to get the data back to the investigators. The timeline for data generation will depend on a number of factors. I am hesitant to even say a timeline, but for example, the cohorts that are going into the pipeline now are expected to finish the pipeline between 4 and 7, in the next 4 and 7 months.
Okay. And So is it expected that we will have capacities to receive BAM files? We do. I just want to make sure if I have to state that on the application, that's an important thing or not. We weren't sure.
Could you speak up again? I missed a couple words in there.
So,
could you repeat the question?
Sure. So we want to know if we have to put in the application that we have space to receive BAM files, for example, here. We weren't sure if we're going to receive those files or if we're going to access them remotely from here.
Yes, you will need to talk about your capacity at your institution to store those BAM files.
Okay, great. Thanks.
Yeah, so that's a good point. One thing as well is that once you are selected for this program and assigned to a sequencing center, there's a lot of room to collaborate with the sequencing center on analyses. Again, in your application, you're going to have to tell us what resources you have, but just know that this is a partnership, it's a collaboration. There are some really great resources at the state-of-the-art sequencing centers that we're working with, from everything to analysis, well, I guess mostly that, but so, we're really excited about working with Hudson Alpha, St. Jude, and the Broad. So you can keep that in mind, but don't forget to have an analysis plan. Well, it doesn't sound like we have any other questions, so I think we're going to go ahead and wrap this up, but please continue to ask questions to us through all of our links and resources and email addresses. Again, this webinar will be posted on our website, and the slides will be posted for the links and information as well. So I'm going to mute myself now, and then I'll stop the webinar. Thank you.
Thank you.